Hello everybody. Today we are going to explain immune response mechanisms part 2, antigen presentation and processing, mechanisms of lymphocyte activation. When cells of the immune system encounter an antigen, a humoral immune response or cellular immune response or both may occur. Humoral immunity is mediated by B cells, which after stimulation proliferate and differentiate into antibody-producing plasma cells. Cellular immunity is mediated by T cells, which become activated to secrete a number of substances important in the immune response. They also kill virus-infected and malignant cells directly, fulfilling the role of T cytotoxic lymphocyte or killer T cells. Enhancing and supplementing the activity of B cells are another set of T cells. These T cells help B cells to proliferate and secrete antibodies and are popularly known as T helper cells. It is therefore obvious that T cells and B cells need to communicate and interact with each other and with other antigen-presenting cells. This they do through receptors, soluble factors, and various cell adhesion molecules. Antigen presentation, mechanisms of lymphocyte activation, T cell activation, activation of B cells, activation either in response to T-dependent antigens or T-independent antigens. First, antigen presentation. T cells and B cells recognize different parts of an antigen. If a hapten is coupled with a carrier particle to yield an immunogen, B cells react with the hapten to produce hapten-specific antibodies, whereas T cells respond predominantly to parts of the carrier. Presentation of antigen to T cells differ from presentation of antigen to B cells molecule. B cells can recognize native antigens and modified antigens, and this antigen need to be presented in conjunction with cellular MHC antigens. Though B cells do require T cell help for an adequate antibody response and for memory cells to be generated. In contrast, T cells do not recognize native antigen. Antigen must first be processed by an antigen-presenting cell and then presented in association with MHC class 1 or class 2 molecules. In other words, T cells are blind to native antigen and will see antigen only when processed and presented together with an MHC molecule. It has been discussed in previous chapters that CD positive T cells recognize antigen association with an MHC class 1 molecule, and CD4 positive T cell recognizes antigen in conjunction with an MHC class 2 molecule. More often than not, CD4 positive T cells act as helper cells and CD8 positive T cells act as cytotoxic cells, though this is not a rigid rule, as we shall see in the following sections. The pathways of antigen processing. Macrophages were the first accessory or antigen-presenting cells to be identified. Later, dendritic cells, specialized cells found in the lymph nodes and spleen, and B cells themselves were added to the principal list of potential antigen-presenting cells. Several antigen-presenting cells do more than simply capture antigen and display it on their surface. They break down antigen into peptide fragments before presenting it, obscuring its shape and leaving only its distinctive amino acid sequence as the target, a process that requires both time and energy. This distinctive target peptide fragment processed within the antigen-presenting cells is then transported to the surface of the cell and displayed in the antigen-binding cleft or groove status, an integral part of both MHC class 1 and class 2 molecules. Here are the two antigen processing pathways. Pathway A for exogenous antigens that is presented on surface association with MHC class II antigens, and pathway B for endogenous antigens presented in association with MHC class I antigen. Pathway A, exogenous antigens. Pathway A describes the events that take place when the antigen concerned is freely circulated foreign material not found intracellular or bound to cell surface. Such an antigen has been termed exogenous antigen and is taken up by specific classes of antigen presenting cells such as B cells, macrophages, or dendritic cells that are specialized for processing exogenous antigen. The uptake of these antigens is either by receptor-mediated endocytosis or as a result of simple pinocytosis. The antigen now lies in an endosome within the cytosol of the cell. In the endosome, under the influence of an acid pH, caffeine-like proteases denature or fragment the origin protein molecule. The peptide fragments then bind within the cleft of the MHC class II molecules, which are also found in the cytosol of the cell. The MHC class II antigen complex is then transported to the cell surface in vesicles and displayed to the exterior. Such an MHC class II antigen complex is recognized preferentially by the CD4 positive T cells, and T cell help for antibody production is on its way. The remaining peptides of the original protein antigen go into a lysosomal compartment for complete degradation. Pathway B, intracellular, such as viral infection or malignant change. These antigens are known as endogenous antigens, being internal to the body's own cells. Processing of endogenous antigens occurs in the Golgi complex, an area specialized for dealing with improperly folded proteins synthesized within the cell. Such internally synthesized proteins need to be presented to the second major class of T cells, the cytotoxic T cells. Endogenous antigen is denatured or fragmented in the Golgi compartment, where they come into contact with MHC class I molecules. The target peptide fragment binds to the antigen binding groove on MHC class I molecules and is transported to the exterior of the cell membrane. Such a peptide fragment bound to the MHC class I molecule is preferentially recognized by the cytotoxic T cell, and the killer cytotoxic T cells has therefore been targeted onto the apparent cell. However, some proteins encoded by the genes of an infecting virus are synthesized in the cytosol. How does the cell get them into the Golgi compartment? This is achieved by specialized transport proteins called TAB, or transporter associated with the antigen processing proteins. Viral proteins in the cytosol are degraded by proteasomes into viral peptides. The peptides are picked up by TAB proteins embedded in the membrane of the endoplasmic reticulum. Using the energy of ATP, the peptides are pumped into the lumen of the endoplasmic reticulum, where they assemble with MHC class I molecule as described above. This complex then moves through Golgi apparatus and is transported to the exterior of the cell membrane. Such a peptide fragment bound to the MHC class I molecule is also preferentially recognized by the cytotoxic T cell. Understanding the phenomenon of antigen processing has helped investigators manipulate the immune system for clinical purposes. Traditionally, vaccines have consisted of whole pathogenic organisms, live or killed, or a protein extract such as a toxin. For some diseases such as malaria, this is not feasible. Besides, whole cell vaccines may have risky side effects. Vaccine scientists are now trying to design synthetic peptides representing a small fraction of the actual antigen to trigger an immune response. To do so, these peptides must stimulate both helper and cytotoxic T cells and bind to MHC molecules. MHC-based technology should also make it possible to develop compounds that bind very strongly to those MHC molecules that bind self-antigens and cause disease. As an autoimmune-related disease states, by blocking the binding of self-antigens on MHC molecules, such compounds might suppress the autoimmune response. Hence, some of the immune system's own precision and power can be directed to fight against disease. Mechanisms of lymphocyte activation. First, T cell activation. The first event that triggers T cell activation is T cell binding to antigen presented in conjunction with MHC molecules. The antigen binding cleft on MHC molecules shows variability, thus supporting the theory that they were made to bind foreign peptides. The T cell receptor CD3 complex therefore interacts with foreign peptide. Simultaneously, the regions of the MHC molecules that display this antigen on the surface of antigen presenting cells interact with the appropriate CD4 or CD8 molecule on the surface of the T cell. This interaction then generates a series of biochemical reactions which are aimed at accomplishing two important events: the secretion of interleukin 2 by the T cell and the expression of receptors for interleukin 2 on T cell surface. Excuse me. When interleukin 2 and its receptor interact on the cell membrane, T cell proliferation occurs. Activated T cells secrete several other lymphokines, including interferon gamma, which help regulate the T cell cycle by the interleukin-interferon circuit. Mechanisms of T cell activation and proliferation. The initial interaction between T cell receptor and antigen stimulates membrane-bound phospholipase C to hydrolyze membrane-bound inositol. This results in generation of two biologically active metabolites, inositol triphosphate and diacylglycerol. The inositol triphosphate increases cytoplasmic free calcium, and diacylglycerol activates the enzyme protein kinase C. These two metabolites are responsible for activating T cells. An activated T cell is then able to transcribe, translate, and express two important gene products, the interleukin 2 molecule and the interleukin 2 receptor. Thus, interleukin 2 is secreted, and the interleukin 2 receptor is expressed on the cell surface. When interleukin 2 and its receptor interact on the cell membrane, T cell proliferation occurs. Here, the cell membrane of the T cell, we see the T cell receptor binding an antigen represented on MHC molecules. This causes an signal to membrane-bound inositol to be to produce the two metabolites, inositol triphosphate and diacylglycerol. This leads to increase in calcium ions and protein kinase C. Both cause transcription of interleukin 2 and interleukin 2 receptor genes together with other lymphokines. Here we see the same process. And we can see the interleukin 2 receptors become expressed on the cell surface, and also a release of interleukin 2 cytokines that cause T cell proliferation. Co-stimulatory signals during T cell activation. Is there any other co-stimulatory signals that aid in this process? Certain cell adhesion molecules have been identified that are necessary for efficient interaction between T lymphocytes and accessory cells or target cells. These co-stimulatory signals involve the interaction of accessory molecules on the antigen-presenting cells with their corresponding ligands on the T lymphocytes. These co-stimulatory molecules are only sensitized when toll-like receptors on antigen-presenting cells bind to pathogen-associated molecular patterns of microbes. Here in this table, we can see cell adhesion molecules on antigen-presenting cells and their ligands on T cells. MHC class 1 peptide with TCR-CD8. MHC class 2 peptide with TCR-CD4. ICAM1 with LFA1. B71 and B72 with CD28. LFA3 with CD2. The importance of lymphocyte function associated LFA1 molecule is evidenced by experiments with monoclonal antibodies which block LFA1. This leads to inhibition of cytotoxic T cell activity, natural killer cell activity, and antibody-dependent cellular cytotoxicity. Patients deficient in this factor suffer life-threatening bacterial and fungal infections. The protein which is believed to be the ligand for LFA1 is the intercellular adhesion molecule 1, ICAM1. Antibodies that block ICAM1 produce the same effects as blockage of LFA1. Various cytokines like interleukin-1 and interferon gamma increase ICAM expression on the B cell surface. The CD2 ligand is the LFA3 molecule blockage. Of these molecules also decrease T cell killing activity by blocking T cell target cell adhesion. Differentiation of T8 lymphocyte into an effector cell, the functioning cytotoxic T lymphocyte. Molecular interaction between the antigen-presenting cells and the T8 lymphocyte produce signals and cytokines for activation of NAV8 lymphocytes. Once activated, signals and cytokines from effector T4 lymphocytes, primarily T helper 1 cells, will then be able to activate T8 lymphocytes. Turn on genes responsible for the proliferation and differentiation of the T8 lymphocyte into an effector cell, the functioning cytotoxic T lymphocyte. Interleukin interferon circuit during T cell activation. Antigens are processed and presented to T cells by antigen-presenting cells such as macrophages. This shifts the resting T lymphocytes from the G0 state to the early G1 phase of the cycle. G1 cells sensitize receptor-inducing factors and other proteins. Under influence of interleukin-1 from the accessory cells, some of the early G1 cells proceed into the late G1 phase of the cell cycle, where interleukin-2 promotes expression of interleukin-2 receptors on late G1 cells. Other lymphocytes moving in a similar cell cycle are stimulated by interleukin-1 to secrete interleukin-2. Interleukin-2 in turn stimulates interleukin-2 receptor-bearing cells to proliferate or actively replicate and enter what is known as the GS synthesis phase in the T cell activation. Actively replicating T lymphocytes secrete interferon gamma. Interferon gamma has a positive feedback effect on interleukin-1 production, antigen processing, and expression of MHC class 2 molecules, thus promoting accessory cell functions, resulting in a cyclical increase of activated T cells, predominantly of the T helper 1 subsets. Here we can see the interleukin interferon circuit, where antigen-presenting cells present antigens to T cells that is still in G0 state. During this presentation, antigen-presenting cells secrete interleukin-1. Interleukin-1 cause the maturation of T cells from G0 to G1. Then G1 cells began to express interleukin 2 receptors. Other G1 cells began to express interleukin 2 cytokines, secrete interleukin 2 cytokines that, when binded interleukin 2 receptors on the T cell surface, leads it to become proliferated, and some of these T cells began to secrete interferon gamma that cause the cyclical effect on antigen presenting cells to release more interleukin 1, so the activation become more and more active. Activating of B cells. Activation of B cells. It is now evident that for many B cells to make antibody to a particular antigen, T cell help is essential. Such antigens are also called T-dependent antigens. There are others, notably the capsule of the organism, Streptococcus pneumoniae, which are T-independent antigens and can evoke antibody production from B cells without T cell help. Activation in response to T-dependent antigens. It has been shown that T cells have first to be activated by separate process which does not involve B cells, as we have just seen in the previous section. Pioneering experiments by A. Murchison in 1971 showed that for helper T cells and B cells to calibrate, they must each in turn recognize parts of the same antigen molecule. This and other experiments have provided compelling evidence that the activated helper T cell needs to be physically close to the B cells. Signals needs to activation of B cells. First signal for the activation of naive B lymphocytes occurs when B cell receptors, the surface immunoglobulin on the surface of the B lymphocytes, binds epitopes of T-dependent antigens having a corresponding shape. A second signal is also needed for the activation of the naive B lymphocyte. This is provided when a component of the complement system C3b binds to the microbial surface. C3b is subsequently degraded into C3d, which in turn binds to the complement receptor called CR2 on the cell surface of B lymphocyte. Here we can see the activation of naive lymphocytes, signal 1 through B cell receptor that recognize the antigen, and signal 2 through C3 complement, C3d, that binds CR receptor 2. Activation of the naive B cell triggers a series of metabolic events in the B cell similar to what has been discussed for T cell. The result is gene activation and increased production of MHC class II molecules, co-stimulatory substances such as B7 and CD14, and receptors for interleukin 2 and interleukins 4 to 6 required for B cell growth and differentiation. T cell B cell interaction. Antigens bound to surface immunoglobulins on the B cell are internalized and degraded to oligopeptides which bind to MHC class II proteins within the cell. The antigen peptide MHC class II complex then goes to the cell surface where it is recognized by antigen specific T cell receptor on the activated helper T cell, primarily T helper 2 cell. The B cell having displayed antigen in conjunction with the MHC class II molecule has now moved from the B0 stage to the B1 stage on its cycle. Here co-stimulatory molecules such as CD28 and CD40L on the T helper 2 cell bind to B7 and CD14 40 molecules on the activated B lymphocytes. As a result of this activated T helper cell B cell interaction, the T helper 2 cells produce cytokines such as interleukin 4, 5, and 6. Collectively, these cytokines enable activated B lymphocytes to proliferate, stimulate activated B lymphocytes to synthesize and secrete antibodies, and promote the differentiation of B lymphocytes into antibody secreting plasma cells. T helper 2 cells also enable B lymphocytes to undergo affinity maturation through somatic hypermutation and to switch antibody classes that is produced either immunoglobulin G, A, or E. This allows the B lymphocytes to fine-tune the shape of the antibody for better fit with the original epitope and to produce the class of the antibody of greatest value for that site. Here the event is involved in B cell activation. First, the B0 cell or B naive cell lymphocytes recognize an antigen and and presented it on MHC class 2 antigens. That is recognized by T cell receptors. T helper cells then become activated and secrete interleukins such as interleukin 4, 5, and 6. Interleukin 4 and 5 cause B cells to move from B1 phase to B2 phase and interleukin 6 move B cells to B3 cells that also differentiate into plasma cells and begin to express and secrete antibodies that fit the previous antigen. Some B cells become memory cells and don't differentiate to plasma cells. B cells move into a B2 stage of activation and display receptors for interleukin 4 and interleukin 5. When the receptors and the corresponding interleukin molecules interact, B cells grow and proliferate, yielding several clones of B3 cells. B3 cells in turn display receptors for interleukin 6 and other related cytokines. Interaction of these cytokines with their receptors on B cell pushes the B cell into the final differentiation stage, the plasma cell, and the antibody production ensues. A set of B3 cells, however, remain unstimulated by interleukin 6 and other related cytokines, albeit with the receptors displayed. This pool of B3 cells constitutes the memory cells. Subsequent exposure of B cells to antigen will yield a much faster and greater antibody response since the terminal differentiation stages before antibody production are all set. To go into production, these B cells only need renewed fueling with interleukin 6 and related cytokines. Other antigens or other agents which induce polyclonal B cell proliferation are Epstein-Barr virus, Chlamydia trachomatis, and lipopolysaccharides which react with non-immunoglobulin receptors on the B0 cell. Activation in response to T-independent antigens. T-independent antigens are usually large carbohydrate and lipid molecules with multiple repeating subunits. B lymphocytes mount an antibody response to T-independent antigens without the requirement of interaction with T4 lymphocytes. Bacterial lipopolysaccharides from the gram-negative cell wall and capsular polysaccharides are examples of T-independent antigens. The resulting antibody molecules are generally of the immunoglobulin M isotype and do not give rise to a memory response. There are two basic types of T-independent antigens, TI1 and TI2. TI1 antigens include lipopolysaccharides from the outer membrane of the gram-negative cell wall and bacterial nucleic acid. These antigens activate B lymphocytes by binding to their specific toll-like receptors rather than to B cell surface immunoglobulin receptors. Antibody molecules generated against TI1 antigens are often called natural antibodies because they are always being made against bacteria present in the body. TI2 antigens, such as capsular polysaccharides, are molecules with multiple repeating subunits. These repeating subunits activate B lymphocytes by simultaneously cross-linking a number of B cell receptors. Thank you.